Stop talking. Stop talking, everybody. Okay. Understand what how all common ancestry of life is linked. What are the special attractions of water molecules? Obviously, the hydrogen bonds. Look at that. Look at the different properties of water molecules, like cohesion, adhesion. What happens when it freezes in terms of expansion, stuff like that. Look at its interactions with hydrophobic substances or hydrophilic substances. Look at carbon in terms of chapter 3. How does carbon interact with other molecules? How many valence electrons does it have? What type of bonds does it form? Why does carbon the backbone of all life? What is the difference between hydrolysis and dehydration synthesis when it comes to building polymers out of monomers? In what situation would you use hydrolysis in terms of like breaking things down or building things up? And which one creates water? This is already being recorded. Which one creates? Yeah, I will do it. Yes. All right. Fantastic. All right. There goes Becky. Anyway, so if I gave you a polysaccharide, let's say 10 sugars long, right? And I gave you the chemical equation of that. Could you tell me how to write the chemical? Or if I told you that there was a polymer that was 10 sugars long, could you write the chemical formula for that? Meaning a link of 10 sugars, how many molecules of water needs to be removed? All right, we're looking at 9 molecules of water would need to be removed, correct? Because how many lines would you draw between 10 circles? It'd be 10 lines, right? And so if I say it's going to be 9 links, right? You would just take away 8 molecules of water. If I said it was 10 links, you would take away 9 molecules of water. So you just subtract that if it was 9 molecules of water that you subtract, that's 18 hydrogens and 9 oxygens, correct? So there's going to be a question very similar to that. If I told you that a specific DNA sample had, let's say, 25% of it was adenine, could you then tell me the other 3 nucleotide bases based on Chargaff's rules? Yeah, very good. If I drew a picture of a specific functional group, of the main functional groups, could you tell me if it was a carbonyl or a carboxyl or an amine functional group, something like that? Know the structural differences between saturated and unsaturated fats. The ones that have, the kinks. The double bonds between the hydrogens is going to give an unsaturated fat. Saturated fats have single bonds between the hydrogens. They're going to be straight. And then obviously, and also tell me how that is important in terms of the fluidity of cell membrane. So when it's cold, we want lots of unsaturated fats, right? And so it kind of allows it at lower temperatures to be very flexible. So you want king. And animals, right? Like fish, for instance, you want cholesterol does the same thing. So cholesterol presence in its cell membrane is going to allow for more flexibility, specifically like organisms that live in really sub-zero or freezing temperatures, things like that. So the cells don't freeze themselves. Their function is specifically doing to the fluid mosaic molecule. It's the fluidity of the cell membrane itself so it doesn't fracture or break. So there's going to be a couple of questions about specific cell organelles, right? Like, like what is the function of a smooth ER versus a rough ER, right? And then what type of cells would you see lots of smooth ER versus cells that would have maybe lots of, lysosomes or something like that, or cells that would have maybe lots of mitochondria. So let's link cell type with cell organelle, and then just basic, cell organelles and their functions. What else? Oh, the endomembrane system, right? Know what are the main components of the endomembrane system, right? Starting from the nucleus itself, and then going all the way out to the plasma membrane, and then the steps of, how that endomembrane system works. Just the endomembrane system, like how proteins are made, and they go through like a rough ER, bloop, come out, go to the Golgi apparatus, get processed, and bloop, come out the Golgi apparatus. If they're meant to be excreted out of the cell, then they're going to fuse with the plasma membrane. Otherwise, they'll stay inside of the cell, and they can maybe possibly be lysosomes, things like that. Okay, so in terms of cellular energetics, understand cellular respiration. What's the final electron acceptor in cellular respiration? What happens in the inner membrane of the mitochondria? In terms of how the electron transport chain and the proteins that are involved are pumping protons in the inner membrane space. And then ultimately, what is the main driving force that powers ATP synthase? Right? It's that proton motive force, that chemiosmosis of protons moving down that gradient, that electrochemical gradient causing ATP to be formed. In terms of photosynthesis, right? What are the main things that are going on in the Calvin cycle, right? So on a general scale, right, are electrons being added? Are electrons being removed? Is there being reduction? Is carbon fixation happening there? Or is carbon being released in the the Calvin cycle? You know, what are the basic things that are going on? What are the molecules that fuel the energy for the Calvin cycles? Since the Calvin cycle is where we produce a 3-carbon sugar, where does that sugar get its energy, right? Right, ADPH and ATP, which are products of the light reactions, correct? So these are basic, fundamental, conceptual questions that you really don't need to understand details for. In terms of protein synthesis, what do we need for transcription to start, right? So what's going on? Think about the promoter, transcription factors, Tata box, things like that. Just basic. What do we need for transcription to start? What's the difference between codon and anticodon? And how does the transfer RNA? What role does it play in that in terms of how the anticodon matches up with the codon? We talked a little bit about that. Let's talk a little bit about integral proteins in the plasma membrane, right? What is the chemical composition of an integral protein, meaning that it has to go all the way through? Parts of it that are sticking outside of the cell, right? Those are going to be hydrophilic, and parts of it that are going to be inside in that phospholipid bilayer are going to be hydrophobic. So any molecule, specifically a protein, that has those two properties, right, is amphipathic. There you go. So those are going to be some. If I showed you a picture of the plasma membrane, right, in terms of its extracellular matrix, phospholipids, and you have chains of polysaccharides that are attached to the lipids, you have glycolipids. If you have chains that are attached to the proteins, you have glycoproteins. And what is the function of that, right, when you have these different polysaccharide chains? It's for communication, it's for cell-to-cell recognition, right? What types of things are going to be able to pass through the cell membrane quickly, right? You got nonpolar, large or small. Oh, so what would be an example of a small nonpolar thing, right? Gases like oxygen, carbon dioxide, things like that. So let's look a little bit about osmosis and diffusion, right? Remember those U-shaped diagrams, those U tubes, and they have that line down the middle, and the left side and the right side. Well, that line is some sort of permeable membrane, right? And so if I give you concentrations of solutes, whether they're sugar or sucrose, glucose, salt, whatever it is, understand that the problem is going to ask you, well, the membrane is permeable to one of the solutes, not both of them. And so those solutes will move down their concentration gradient, right? And so they're going to go from high to low, right? If the membrane allows that to happen, right? If it's permeable. After everything moves based on the permeability of the membrane, then you have to look at the concentration of solutes on both sides of the U-tube, right? Right? In terms of like which has more stuff on either side. The side that has more stuff, right? The side of the tube that has more solutes, a higher concentration of stuff in there. What do we call that in reference to the other side? It's hypertonic, right? And so what direction is water going to move? Into that side. And so there's going to be a couple questions about that. If you have a cell from an organism and you place it into a solution, we're going to use these terms hypertonic, hypotonic based on the fluids on both sides of that cell. On the inside of the cell versus the outside of the cell. Now, if the outside of the cell is super salty, it's hypertonic, it's going to draw water out of the cell, correct? And so the water is going to lose mass. So if I draw you another graph, right? Graph that goes like this, like this, like this, in terms of the mass gain or decrease of a specific cell, you can determine the relative concentration of the inside of a cell and its environment based on whether or not it doesn't gain mass, so it's isotonic, right? So it's going to be the same concentration of the solution that it's in. If it gains mass, then it is hypotonic inside. Oh, sorry, it's hypertonic inside, my bad, because it's gaining mass. So it's in freshwater environment. It's in a hypotonic environment. And so there's going to be a graph that shows that, and you have to tell me, which one has the highest concentration, which one maybe is isotonic based on those graphs. And a U-tube, you'll have to know, what direction is water moving, which one is hypertonic versus the other side, stuff like that. What's different about a steroid hormone versus a peptide hormone in terms of its ability to communicate with the cell? What's unique about a steroid hormone? It's nonpolar, number one, so it can do what? Go right through the membrane, right into the nucleus, and act specifically on the gene. So it might be able to turn on transcription factors by bonding directly into those transcription factors inside of the nucleus. Whereas a peptide signal it's going to have to bond to some sort of receptor protein. Blah, 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 blah. Anyway, so there's that. Okay, let's talk a little bit about enzymes. Okay, what is the main function of an enzyme? And if I drew you a graph, remember those graphs that have... I'm going to draw you a graph real quick. It could be any one of these two graphs. Okay. One of those graphs. See those graphs? And then you might have like little arrows, right? Whereas like this is the reactants, these are the products. and you have little arrows that are like this, or maybe little arrows that go like this, right? Stuff like that. Everybody see that? Whoop, whoop, whoop. See that? Okay. Okay. And can you tell me what all those arrows represent in terms of exergonic and endergonic reactions? You know, what is delta G, right? And how does and what type of reaction is delta G positive and delta G is negative in terms of gives free energy? And so, by the way, let me add another line to these. Look at that. See that other black line that I added, right? That's going to be what happens with that black line? Why is it shorter? Why is that little lump shorter? Yeah, it's catalyzed with an enzymatic reaction, so understand what that does. Speaking of enzymes, think about what makes something a competitive inhibitor and what makes something a non-competitive inhibitor, aka an allosteric inhibitor. And so if I gave you a graph, another graph here, of, I don't know, let's say your basic rate of reaction graph that looks like this. There you go, rate of reaction graph. Whereas, your independent variable there is going to be the, let's say, solute concentration, and the dependent variable there is going to be the rate of reaction. Explain why it plateaus off, and what would be some of the things that you could do to cause the rate of reaction to increase, or in that case, decrease. Think of and remember my situation with the Coachella, whereas the porta potties are the enzymes, and if you have 1 million people, there's only so much urine output, right? And the urine output per second is going to be your rate of reaction. If you can add as many people as you want, right? It's only going to be producing so much urine per second, which is going to be the rate of reaction. What's the best way to increase the amount of urine that's being produced? Add more porta potties, right? Increase what? Concentration. Yeah, all right, exactly. And so there's that. So there was a couple of graphs that we showed you that had to do with maybe different rate of reactions that had to do it in different temperatures and different pH. And so, if you have a high rate of reaction in like really cold temperatures or really hot temperatures, what sort of conditions or what sort of enzymes or what sort of organisms would you expect to see that in? If you saw, rate of reactions in enzymes that were really high and optimal in a low pH versus a high pH, what sort of organisms or what sort of conditions would you expect to see that? Yeah, specifically in humans. When I think about in humans, where would you see a human enzyme that would be highly functional in a very acidic environment? Right, okay, and so think about that and where and why you would see that. And what are the conditions that cause enzymes to no longer function anymore? Why doesn't it function as the pH changes? And understand that, how pH and temperature denatures enzymes by affecting the bonds that hold together its tertiary structure. Things that have a lot of covalent bonds, that specifically disulfide bridges hold together those proteins together really well. Now, when you think of an enzymatic, a metabolic process that has one substrate being turned into another intermediate compound, which gets converted into another intermediate compound, which eventually becomes a product. There's enzymes in between all of those conversions, and so if the production of the product, right, ends up turning off the initial enzyme, that's an example of what type of feedback? A negative feedback loop. So a positive feedback loop, on the other hand, then would be something that as we go further and further away from a stimulus, that we're actually turning on the production that drives us further away from the stimulus. One example of that would be, like pregnancy. And so when we think about the lac operon and the trip operon, how do those work in terms of negative feedback loops? And where one of them, right, you have, let's say the trip operon, where we're producing tryptophan, the production of tryptophan, the presence of tryptophan acts as a co-repressor to turn off the transcription of those genes. Whereas the lac operon is an inducible operon because the presence of lactose doesn't turn off the production of the enzymes. It turns on. It acts as an inducer or an activator to turn on the transcription of the enzymes that eventually make proteins that break down lactose. So know the difference of those two. Which one's inducible, which one's repressible operon, blah, blah, blah. What's the difference between something that is reduced versus something that's oxidized? Leo says GER. Leo says GER. Oil rig is another way to remember it. You guys need to go into your book and look at the action spectrum of photosynthesis and how it reacts with the absorption spectrum and how they're overlap the two, right? The absorption spectrum looks at all the wavelengths that are absorbed by pigments that you'll find in photosynthesis, whether it's chlorophyll A, chlorophyll B, all the accessory pigments. which ones are absorbed the most? What wavelengths of light are absorbed the most? And how does that correlate with the amount of photosynthesis that occurs, right? And so obviously certain wavelengths, I mean, everybody knows that what wavelength is least absorbed by light, right? Green, right? So if I give you a chart, you're gonna have to look at the graph, and you're gonna have to find out what wavelength specifically based on the graph. So you look at the peaks of the graph, right? And then go down and look at the graph's independent variable and say, well, this wavelength based on this graph is absorbed the most. And obviously it correlates with the most photosynthesis. It makes perfect sense, right? Because the more light that you have, the more light reactions. The more light reactions we can have, the more dark reactions we can fuel. The more oxygen is produced or the more carbon dioxide is consumed, however you want to measure photosynthesis. There's a couple diagrams in the book that describes the difference between a C4 plant and a CAM plant. C4 plants and CAM plants have adapted to like hot environments, right? Where they have to pretty much shut down their stomata a lot of the time so that they prevent getting dried out. And so a C4 plant versus a CAM plant, they differ in the sense that a C4 plant has different regions of the leaf in which it stores its organic acids, whereas a CAM plant, it's just the only difference. A CAM plant, it's open at night and closed during the day. And so if I give you a diagram or a picture, could you tell me from that picture whether or not that process is done by a C4 plant or a CAM plant? Got it? Okay, talking about mitosis and genetics now. If I give you pictures of cells that have, let's say 2 chromosomes, n equals 2, could you tell me what phase of mitosis or meiosis it is in? Just from a picture. You'd have to know whether or not we're separated sister chromatids, whether aligning next to homologous pairs. If you're looking at the drawings, right, you should be able to tell if it's prophase 1 or prophase 2 or anaphase 1 or anaphase 2. So there are 2 diagrams in your book. One is pictures of the nucleus of cells as they go through meiosis. Another one is pictures of the cells as they go through mitosis. You also did that mitosis and meiosis modeling activity in my class so that you could be able to stare at a nucleus of a cell and tell me what phase it's in. Got it? There was also that graph that showed a line going up, plateauing, going down, plateauing, going up, plateauing, whereas the increase and decrease was the amount of DNA in the nucleus, right? And it had the different stages of that graph, and it was correlating with specific stages of the cell cycle. So remember that graph, right? It's coming back. And if I tell you different cell types and how much DNA is in their nucleus at specific stages of the cell, could you tell me what type of cell that is? Whether it's a cell that divides a lot, whether it's a gamete, whether it's a cell that doesn't divide a lot, based on the information of how much DNA it's inside of that cell. Going back to the drawings of what happens in meiosis, understand what are the main events of all the stages of meiosis. In terms of, this is where chromosomes are formed. This is where they overlap and crossing over occurs, right? Prophase. This is where the chromatids pull apart versus the homologous pairs pull apart. The difference between anaphase 1 and anaphase 2. Things like that. If I gave you a bar graph telling me the amount of relative DNA that's inside of specific cells, whether it's like, this much, double that, or double that, could you tell me what type of cell it is? Whether it's a normal body cell, whether it's a normal body cell after duplication, or whether it's a gamete. Know the difference between a diploid cell and a haploid cell. And the fact that a diploid cell has twice the DNA than a haploid cell, right? A diploid cell after DNA synthesis has even twice the amount of DNA of a diploid cell and 4 times that of a gamete. Okay, so just think about that conceptually. It's pretty basic. Moving on to genetics, if I give you a specific long genotype, big A, little b, big C, big D, little e, big F, little f, could you tell me how many gametes, how many different types or unique gametes can that organism produce? You can write them out, or you can look at each individual genotype, right? Big A is only going to produce one type of gamete. It has a big A. But if it has a big C, little c, there's 2 different types of gametes. You can have a gamete that has a big C or a gamete with a little c. And so make sure that you understand how to use the rules of probability to be able to look at what are the different unique types of gametes that can be produced. But also, if I gave you 2 parental genotypes, could you tell me what the probability of their offspring's genotypes are or offspring's phenotypes are? So if I give you like a big B, big T, little t, crossed with a big B, little t, could you easily tell me, well, how many of the offspring are going to be tall or black and tall? And so it's just use the rules of probability to do that. So that's going to be quite a few of those questions there. Another difference between Sex-linked, yes. There's going to be a question that you're going to have to actually do a sex-linked cross. So understand, like, first of all, sex-linked traits like with cats and X inactivation, right? Where we have those cats that are calico cats, black and orange, is because you have two X's in the female cats, and there's two genes. There's a black gene and there's an orange gene, right? And they're located on the X chromosome or allele. There's one gene. And with bar bodies, is one of the chromosomes become so overly methylized that they kind of form this bar body, and they can become inactivated. And so if that's the orange one, then the black, and they have black, then that patch of skin is going to be black. So understand that only females can have that condition, right? So when you do a sex-linked cross, you set it up like this: XX and XY on the side. And so there's going to be two questions about sex-linked crosses. And so if you know what the dad is and you know what the mom is, could you do a sex-linked cross and tell me the percentage of offspring, whether it's male or female, that are either going to have this sex-linked trait or not? Can you perform a simple sex-linked trait? And understanding other types of things, like what's epistasis, where one gene affects another gene, right? Like with albinism or albinism. Yeah, so like how one gene, right? You can have a gene for the color of skin of a rat, whether it's brown or black. So let's say it has but this other gene over here is going to be like whether it actually has pigment or not. So you have a white rat that's white, not because of this gene over here, but because of this gene over here. So this gene, the production of pigment at all, is influencing another gene. The opposite of that, I guess, would be not the opposite of that. Another form of how complex interactions of genes would be, let's say, pleiotropy, where a pleiotropy is where you have one genetic mutation affecting many different phenotypes, right? So you look at achondroplasia or Marfan syndrome, where you have these conditions where you have one genetic mutation and a lot of different body systems are affected. You could have, cystic fibrosis is not cystic fibrosis could be an example of that, because you have one genetic mutation and you're having multiple different phenotypic problems with that. The opposite of that, I guess, the opposite of like one genetic mutation, many phenotypes, is polygenetic, right? Where you have like many different genes working on one phenotype. So it would be like skin color, height, weight, where things fall on like that normal distribution term. Understand how to do crosses of either codominance or incomplete dominance, where the heterozygote is going to be a totally different phenotype. Whether you have red flowers and white flowers give you pink flowers. It's the same thing with codominance. You got a red cow and a white cow gives you roan cows. The only difference is that there's individual red and individual white, but the Punnett squares are pretty much the same. So you're not going to get any problems with that. Pedigrees, no. Probably should be, but thank God. Sorry. I'm going to add one right now. Okay. Understand how is crossing over and gene linkage related? In terms of like, what is gene linkage, right? Gene linkage is two genes on the same chromosome, right? So the closer those genes are on the same chromosome, the more often or least often they're gonna cross over and separate, right? Think about that. That was the whole purpose of a little meiosis, where we were looking at the patterns of the Sardaria, the fungus, right? That had two black and then two tan, things like that. Transformation of bacteria, right? What is transformation of bacteria? If I give you a scenario where you have like ampicillin resistant, non-ampicillin resistant bacteria and a bunch of different plates, could you tell me what should grow on what plate? Things like that. In terms of DNA replication, understand why DNA replication occurs in the 5' to 3' direction or vice versa, right? However DNA replicates. Understand that we can only add nucleotides to the 3' end of another nucleotide, right? What is the 3'? It's that third carbon in that nucleotide's sugar. What are the roles of all the enzymes involved in DNA replication? Helicase, DNA polymerase 1, DNA polymerase 3, RNA primase, DNA ligase, things like that. What are their jobs? What do they specifically do, right? And blah, blah, blah. If I gave you a codon chart, the big square thing that had like first base and had all the little letters, the codons, could you then use that codon chart to be able to understand how protein synthesis works? If I gave you a DNA sequence, could you then transcribe that into a messenger RNA sequence and then tell me what the amino acid sequence would then be? Going in that direction, or could you then work yourself backwards? Yeah, so if I gave you like a DNA of AAA, then the messenger RNA would be UUU, and then the messenger RNA is what you then use the codon chart for. And then UUU is, I don't know what is UUU, phenolphthalein or something like that. Phenylalanine, sorry, not phenolphthalein. That is an indicator. So there's that. Epigenetics. What are some of those 7 steps? Acetylation, methylation, acetylation, messenger RNA degradation. which one of those things falls under the category of epigenetics or the phenomenon of epigenetics. You don't need to know like anything in huge detail, but which one of these processes would fall under the understanding of how, even though two individuals can have the same genotype, they're going to have different expressions of their phenotype based on all the different things that could possibly be influenced by epigenetics. Which one of those would be the most damaging? Right, something that happens in the beginning of a gene, right, and it's a deletion. Got it? Towards the beginning of a gene, and it's deletion, which causes shift. Particularly, which it would cause an early stop codon. Those are the most harmful. In terms of plants, there's maybe like, 3 questions? Yeah, like what is auxin in charge of, right? What does ethylene do? What does abscisic acid? Just basic main function, right? What would cause a plant to grow towards the light? Basic. Remember the POGL that we did, right? How we have certain things building up on certain sides of the cell, causing those cells to elongate, which would then cause that. Okay, understand that phenomenon. Once again, what's the difference between a short day and a long day plant? And what would trigger the blooming of that? Right? A short day plant blooms when there's a long night or short day. And a long day plant is when there's a short night and a long day, right? So there's this critical period of light, right, that's going to cause that. Question on that. Obviously, how positive and negative feedback differ. A little bit about immunity, right? What is the difference between active and passive immunity? Can you give me examples of both? What's cell-mediated? What's an example of cell-mediated immunity versus humoral immunity? What's the role of the MHC, right? The major histocompatibility, right? Those proteins in terms of how do they engage with T cells? What's the overall function of that? Well, what is a bacteriophage? What does it do? There you go. That's all you need to know. What type of immunity is that? What main category would that fall under? What is the difference between non-specific and right? One's innate, right? The other one is learned, like acquired, exactly. In terms of the nervous system, just what's your basic anatomy of a neuron? Where does it receive information? Where does it deliver information? What's the gap in between those informations? And basically, what are the main steps of action potential that cause an action a neuron to fire? And could you put them in order if I give it to you, right? I mean, no, I'm getting a lot of this from you, Strauss. No, I don't know that. No, you did a horrible job teaching me that. No, what's the operation? What is the role of the sodium potassium pump into all that? Right, in establishing a membrane potential, right, in the firing of membrane potential. We have sodium and salty banana. Think of salty banana. Ecology. If I gave you a food web, could you tell me which one's the producer, primary consumer, secondary consumer, things like that? You remember, there were some population problems where you had to know, like, what is the formula that would give you logistical growth versus exponential growth? And if I gave you a population and told you the birth rate and the death rate, could you tell me how many individuals were added year from year? Very simple. Birth rate tells you how many people are added every year. The death rate tells you how many people are subtracted. So if you want to know how many people are added to the overall population every year, you got to take the 2 and minus it. How many people were born? How many people were killed? Right. So the birth rate is very simple. If the birth rate is like 0.4 or 0.04, that's the percentage of individuals of the main population that were born based on the original population. So birth rate, right, is multiplied by the population, whatever the population starts at. A death rate is the same thing. It's a frequency. Like 0.04 is a birth rate, is a death rate. 4% death rate or 4% birth rate. 0.4 would be 40%, right? And so you have to combine this. So there's going to be a problem. Looks similar to that. From individual to biosphere, right? Understand how we organize the fields of ecology. That's a pretty no-brainer. What types of biomes are you going to see the least amount of biodiversity, the most amount of biodiversity? What are the main characteristics of the main biomes? Just the ones that you see right there. There's a question or two on that. And then, what's the effect of greenhouse effect, eutrophication, acid rain, and the ozone layer in one sentence? Like, what are those things? How are they caused? In a basic nutshell, right? Do you have to know the nitrogen cycle? No. But what are the roles of detrivores in the ecosystem? Yeah. Versus producers versus consumers. Like, Nitrogen fixation, where bacteria takes nitrogen gas and puts it into nitrogen-based compounds that organisms can, like plants, can take in. That's it. That's all you need to know, right? What organisms do that? Well, what do detrivores do, right? Know that. There may be one question about magnification like that. It may be having to do with DDT. And so as DDT is sprayed into like your crops, then you have insects that eat DDT, so they build it up. Then you have birds that eat the insects, so that builds up in the birds. And then if we eat the bird, as it builds up through the food chain, that's magnification. You see that as chemicals like that work their way up through the food chain, they build up in concentration. Because you spray a little bit on the plants, but guess what? Insects are eating plants all day long, so that stuff gets built up in the insect. And then a bird eats tons of insects, so it builds up an even higher concentration in the bird. And then by the time it gets to the wolf or whatever the hell, somebody's dying. Because the concentration of the DDT was harmless as you spray it on the plant. By the time it works its way up the food chain, it increases in magnification. So know that concept. And we're done.